Let's take a look at what you're doing this week in lab. We're looking at Module 4, which is microscopy. Your overview will give you a brief description, your weekly objectives, and your weekly activities. You don't have a lot of activities this week, but they do take some time. You have your weekly lecture in the video. You also have some assigned reading, Chapter 10.2, Cell Cycle in the online textbook. You have an interactive here, and then you also have a video on the phases of mitosis. That looks at the content for this week. Then for your science skills, you have several links here on how to use microscopes. And then you also have some resources that are specific for your assignments. And you definitely want to take a look at this one here because it will show you this database that you're going to be using for the assignment. It's not super intuitive. I would definitely make sure that you go through this tutorial before you try to do this assignment too. Your first assignment is to go through this Labster virtual lab on microscopy. Remember, as you go through the Labster, you want to hit this play button up here because that'll allow you to control it and you to answer the questions where you will get a score. If you click on this video here, someone's running all the controls for you, but you're not actually answering those questions. And so that score won't go over into Canvas. As we've done with the other Labster activities, you will also write a reflection answering these questions about the activity in Labster. After you do your Labster activity, you have your assignment 2 for week 4. And this is where you will be using the Allen database to count cells in different phases. You can read through the overview up here. The instructions are given in steps. We had that original tutorial video I mentioned a minute ago. Make sure you go through that. That's going to guide how you're going to do this lab. You're going to be making a poster of this like you've done in the last few weeks. One of the first things you're going to do is to construct a hypothesis. You're going to be counting cells in different phases of mitosis. Your hypothesis is going to have you predict what you think you're going to find. So if I count, say, 100 cells, what percentage of those cells do you think will be in these different stages? Interphase, prophase, metaphase, anaphase, telophase. What percentage of cells do you think will be in each of those stages and why? You want to put your hypothesis out there before you collect the data because that's what scientists do. We don't collect the data and then form a hypothesis. It's perfectly fine if you reject your hypothesis at the end, but we want to first form that hypothesis. You're going to download this spreadsheet. As the tutorial shows, the spreadsheet is going to have in the first column links to different images. Across the top, you're going to have those different stages of mitosis. For each of those images, you're going to count how many cells are in each of those stages. Fill out that entire table, that entire spreadsheet. Here we have screenshots of some of the images that she talks about in the tutorial video. You're going to do this for all of those images that are in that spreadsheet. It's also asking you to save a screenshot of cells that you have found for each stage. So you're not having to do a screenshot of all of the different things you see. Just make sure you include a screenshot of what you think is interphase, prophase, metaphase, anaphase, and telophase. Create at least one image for each of those. You're going to count those cells. You're going to add up the total number of cells that you've counted and how many are in each of these stages. And you're going to convert that to a percentage. Then you're going to create a graph that looks at the percentage of the cells in each of those stages. This is going to be from all of those different images that you have, all the cells you've counted, what percentage are in those different phases. You're probably going to be looking at a bar graph. If you're doing percentages, you actually could look at a pie chart. We don't see pie charts being used a whole lot in biology, but for something like this where you're looking at the percentage of a whole unit, it is valid to use a pie chart. And then what it's asking you to do is to create a second graph where you exclude interphase. What is the percentage of all of the mitotic stages? Remember, interphase happens before mitosis, and then mitosis actually includes prophase, metaphase, anaphase, and telophase. Then you want to go through and answer these discussion questions. What I recommend you doing on your poster is don't number your discussion, because if you read a scientific paper, you don't see people putting 1, 2, 3, 4, 5 on their discussion, right? right? They write it as an essay, as a paragraph formatted essay. And so what you want to do is include this information, but write it like you would a scientific paper, not here's my answers to these questions. Don't repeat the questions here, but make sure that you answer them, but answer them in a paragraph format. And because this is a poster, it is okay in your discussion to use bullet points, because for posters, we do want them to look a little different than a scientific paper, because we want someone to be able to walk up to your poster and that information to kind of jump out at them. So it is okay to put some bullet points on your poster, but you still want to make sure that each of your bulleted points is a complete sentence. It makes sense. You're making it in a format that someone who's not familiar with these discussion questions or with your research can understand that. What I'm seeing from some of the submissions is that the audience needs to understand what you did because you're not giving them enough information to go on. This poster needs to stand alone. Your audience can interpret exactly what you did. Make sure that you are giving enough information to your audience. What it's asking you to do is to take your posters that you've been adding to. So you had a poster from week 1, week 2, week 3. You're going to take those, and so you have basically right now a PowerPoint that has 3 slides in it. You're going to add another slide, so just copy one of those slides and add the information for this week. And you're going to have those same sections. So if I put comments on your previous posters, you might want to go back and look at those comments if you've lost any points so that you get a higher score. Down here, you have the grading rubric. You will be submitting a PowerPoint that this time has 4 slides in it, 1 for each of the 4 weeks so far. The 4th slide, of course, is the one that I'm grading this week. What I'll do next is to show you how you would create a pie chart or a bar graph of your data using Excel. So I've opened that downloadable Excel file that has these links for the images. I'm going to click here, Enable Editing. When you click on these images, it's going to open up that image in the database, and you're going to count. I'm just going to make up some numbers so I can show you how you would do this. What I did is I just put some fake numbers in here. I don't want to go all the way through, so I just put another line in here so I can just illustrate how you would do the graph. I'm going to total how many cells of all of my different images I looked at are in each of these stages. If I click on this cell, I can come up here to function. Right here it pops up with sum. So I can say sum and then say OK. What it's doing is automatically putting in here B2 to B5, which is what I want. That's B2 through B5, so that's good. And say OK. And as we saw before, Excel's great for this because you don't have to type that in anymore. I can just grab this right hand bottom corner here and drag it across, and it'll add my other columns for me. What I also can do now is look at what is the total number of cells of all of these that I've seen. Well, it's actually the sum of this row right here. Let's come in here, and I'm also going to sum this row. Function, and it's going to be sum, okay. But now I'm going to sum across there, okay. I've counted a total of 80. Now let me convert this to a percentage. Percent down here, what would this percent be? Well, it's going to be equal to 61. So I put in an equal sign. I click on 61, divide by 80, because that's my total. I'm going to put a parentheses there and say times 100 to make it a percentage. And I have to close that parentheses there. Take this number, divide it by 80, because that was my total, times 100. That's going to give me my percent. 76.25% were in interface. I can grab the corner and copy it over. And if you come up here, if we do this and sum them, it sums up to 100, which is great because we're doing a percentage. These are the numbers that I would want to put in my graph. Highlight these numbers, insert. So let's do an insert. And let's look at a pie chart first. So let's do a pie chart. So I don't really like the fact that it says 1, 2, 3, 4, 5, 6 down here. What I want it to do is to give me the names of those phases. What I've done is I've copied the values from here into here. So I just did a Control C, and then I came over here and I did a Control V. Now notice it comes up and it says value. That's because these are calculations. But if I come over here to this little control and I do a drop down and say paste values, what I'm telling Excel to do is to give me the numbers from over there. there don't give me the formulas. So now it's giving me the numbers. I just copied this as well. So I did a Control C and a Control V over here. Now, if I grab these data and I go in and do my pie chart, the names are down here as well. That makes more sense than 1 through 6. You want your reader to immediately look at it and see. You don't want them to have to hunt for what does number 1 mean. Now, the other thing we could put on here are the actual percentages. If we click inside of our chart here, so I've got it all highlighted, and then I do add chart element, one of the things we can put on there is data labels. And so let's go ahead and put data labels on, let's say, the outside. So now we know that this big blue one is 76.25. We know that that one is 6.25. So that helps us see that a little bit better. That's how you do a pie chart. If you wanted to do this as a bar chart, you basically could do the same thing. Let's grab these values, insert bar chart, come here, add our chart elements, add an axis title on the vertical. We want it to say percent cells. And those are the percentage of cells that are in each of the phases. This is what it would look like if we had interphase in there. Now, if we leave interphase out, we would have to recalculate our percentages and create new percentages of just based on these columns. And then we could make the graph using the same protocol. That would be how you would make one of those two different types of graphs to show the percentage of the cells in each of those different phases. Make sure that you have 2 graphs on your poster, not these 2 with the same data in it. You could choose whether you want the bar graph or the pie chart. But you're going to have the one including interphase, and then we're going to have one where we exclude all of these cells and we're only really looking at this part of the pie, but then we're looking at that as the whole. If I only count these cells and forget about those, what's my new percentage for prophase, prometaphase, metaphase, anaphase, and telophase? And you can see on this graph, it would make it a lot easier to see what was going on. The fact that this is such a high bar makes it hard for us to see the differences in these numbers here. If I get rid of this one, these will pop up and I'll be able to see those differences a little bit better. And the same thing here. It's a little hard for us to see the difference between the 3.75 and the 5, but if this is taking into account the entire circle, we would see that a little bit better. So that's one of the reasons for leaving interphase out, is to say, okay, when it's in cell division, how much time do these stages take? So you'll be uploading your PowerPoint slide for that. Make sure that you take a look at the grading rubric. And that's everything for this week. If you have any questions on how to use the spreadsheet, the cell viewer, anything from Labster, please don't hesitate to reach out and ask me.